Good morning. This is episode 4 of the Antimicrobial Resistance Talk, or in short, the AMR Talks, where we are bridging gaps and sharing solutions. This podcast is an initiative by the AMR Research and Work on Innovation and Sustainable Solutions Research, also known as the AMR WISE Research Group from University Malaya. Our aim is to increase awareness about antimicrobial resistance, or in short, AMR. AMR is like an intricate puzzle. It's a phenomena where microbes, those tiny organisms, invisible to the naked eye, are evolving to resist the very medications designed to eliminate them. And as these microscopic battles unfold, our arsenal against infection is shrinking, posing a profound threat to modern medicine as we know it. But fear not. Today, we're not just diving into the research of AMR. We are exploring the resilience, the innovation, and the collective determination to rewrite the future of healthcare. In this episode, we have Dr. Yukihiro Akeda. Dr. Yukihiro Akeda is the director of the Department of Bacteriology in the National Institute of Infectious Diseases, Japan. With over 30 years of experience at the Osaka University, Yale University, and NIID, he has been working in the field of bacterial pathogenesis, molecular epidemiology, vaccinology, international collaborative research, clinical research, the development of diagnostic kits, antimicrobial resistance against broad range of bacterial infections such as diarrhea, respiratory infections, systemic infections, and sexually transmitted infections. Wow, that was a mouthful to say. Seems like Dr. Akeda has done a lot of research and contribution to AMR. Thank you for joining this podcast with us. We are very excited to learn from you today.
Okay, thank you.
So maybe you can tell us a little bit of the AMR scenario in Japan.
Yes, in talking about the AMR in Japan, it's same as the other countries in the world. However, the situation of the AMR, it's a bit different when we compare to the situation outside Japan, because as an example, the drug resistance rate of the certain bacteria such as Acinetobacter resistance rate is less than 0.5%. However,
Wow.
when look at the other countries in the Southeast Asian countries, it's around 70 or 80%. So it's totally different, but still we are really considering about the AMR problem.
All right. You would like to tell us about your department. I
Okay.
hear you're in the Department of Bacteriology 1?
Yes. In my department, there are 6 laboratories. Laboratory 1 is for the enteric pathogens such as like diarrhegenic E. coli and Campylobacter. And laboratory 2 is for Salmonella, Vibrio, Shigella, and the laboratory 3 is for the Pneumococci, Legionella, and Streptococci. And laboratory 4 is for the Spirosis, Borrelia, and Yersinia pestis. And laboratory 5 is for the meningococcal disease and STI, such as like gonorrhea, syphilis. And laboratory 6 is for oral bacterial. So like Streptococci and Treponema and Porphyromonas, those kind of stuff. We are working on that.
That's a very good contribution to research. It's such a diverse range of
Yes.
things you're working on.
Mm-hmm.
Okay, so since you're working on all these organisms, I'm sure that you have a lot of molecular techniques that you implement. So in terms of molecular techniques in the fight against AMR, could you maybe explain a little bit on that?
Yeah, the molecular techniques is like whole genome sequencing can tell us a lot about the pathogen itself, and also the not only the phenotypic characteristics of the pathogen. We can see the more information, which kind of the genes inside the bacteria, and also from the gene information, we can speculate which kind of the drug resistance they have, and also we can use those information how they transmit to the other area or countries. So the whole genome sequence data tell us a lot. And also using those information, we can develop the test kit, rapid test kit, using the PCR or other method. So that information is really effective to establish effective tool for AMR control.
That's very interesting. So maybe you could share some examples of ongoing projects related to guiding antibiotic treatment.
Yeah, so the first thing we have to do is we should know the baseline data of the target AMR pathogen. Otherwise, we don't know how we can act on those specific AMR pathogen. So that's why we conducting the genomic surveillance for various kinds of the AMR pathogen. Then once we got the baseline data in terms of the whole genome sequence data, we will design how we can contribute to control the AMR transmission or spreading in the area. And also those data would be very useful, not only in the Japanese people, maybe other countries can utilize those information.
Definitely. Are there any challenges that come with this? It's great work, but what are the challenges that you face?
Yeah, we could say fundings. It's really challenging because when you say the whole genome sequencing, it's much cheaper compared to the several years ago. But still, when you do a lot of the sequencing or the bacterial genomes, it costs a lot. So that is one challenges we have to make some solution. And also another thing is the collaboration with the clinical institute. Some of the hospital is too busy to provide the isolate of the target AMR pathogen. So we should have a good collaboration. Yeah, that's another thing we have to work on pretty well.
So it's definitely, there needs to be a collaboration between the clinical side and the research side to combat AMR.
Yes.
Yeah. Is there any advice you would say on how to foster maybe good collaboration?
Yeah, be nice.
Be nice.
Be nice to the others. Yeah, and also you can tell them or tell other people you like to join to the research group or project. Telling them how these projects are going to be successful, and if the established data is really useful, it can be useful for not only in the research as well as the clinical side. Then you may make them helpful for you.
Definitely, yes. All right. So one of the things you mentioned about your projects is a rapid diagnostic kit. So rapid diagnostic kits, what I understand is they give us answers in very short duration of time.
Yes, that helps a lot to the clinicians when they make a treatment course. So if you know the pathogen is in the patient, you can take accurate action, and also if you know the susceptibility of the pathogen, you can choose the right drugs for that. So rapid test kit will the doctors a lot, and also the and as well as a patient will be helped a lot. Yeah, and also in terms of the nosocomial infection, rapid test kit can provide a quick action against the infection control in the hospital. That also help the other people working in the hospital too. So yeah, rapid test kit are really useful. But it should be cheap, otherwise nobody uses it. Just not only the tertiary hospital, private clinics needs a quick answer of the diagnosis because the patient is not so patient to be stay in the clinic like a couple hours. It's unbelievable. Yeah. And so if the doctor knows a quick answer from the rapid test kit, patient can get the appropriate treatment and can go home much earlier.
Definitely, yes. But are there any challenges that come with this rapid diagnostic test?
Yes, rapid diagnostic kit is usually based on the genomic sequence data. So if the bacteria evolve into another type of another type, so you have to use another genomic information for the kit. So yeah, that's one of the challenge. So only one kit cannot apply all the bacteria as well. So we could have another another type of the kit can cover the all or broader range of the AMR pathogen. So, but still, some researchers and the companies working on that, but still it's not available. So that's another the big challenge in the future.
I see.
But now you can use AI.
Yes.
Yeah, so that maybe help a lot. Just taking micrographs and then apply the pictures to the AI. Maybe AI could tell the diagnosis.
Wow.
We don't know, but it's really simple and it's quick and it should be very cheap. So it's one of the ideal diagnosis kit in the future, I think.
All right, that's very interesting. Future of diagnostics, and with the big help of AI.
Yes, I think so.
You can see that happening.
Yeah, but the AI should be very clever and smart.
I think it's already getting very clever right now.
Yeah.
All right. So Dr. Akeda used to be a lecturer, and now he is heading the Department of Bacteriology 1 in the Research Institute. So you've made a transition from being a lecturer to a role in a research institute. Maybe you can tell me what are the key differences and similarities when you're fostering awareness through lecturing or actively contributing to AMR research. Is there any difference?
Yeah, there is a big difference, and also in some part there is not a big difference. So at the director position, so sometimes it's beyond your specialty. So, but you have to work on that. That's one of the things. One of the difference compared to the previous position. Because when I was in the university, I just keep an eye on the small laboratory. So it's really big difference. And also we have to think about the bigger view of the research or public health program. So because now I have a big responsibility to control and to it's to control the infectious diseases. So not only your specific interest on the project, you have to think about public or country or global health. That's a big difference. However, the similar things, even in your director or the lecturer, there is your passion to the science or other medicine. That's always not changed. And I still feeling like that. It's not changed.
That's great. But talking about research, why do you think research is particularly crucial? So the clinical side, they can play a role in combating AMR, for example, antimicrobial stewardship programs in hospital. So how is research particularly important in combating AMR?
Yeah, without the research, you cannot get the evidence, scientific evidence to support the policy or intervention. So the research is really essential. So without the research, it's just your idea without any supporting data. So the research is essential.
So we can say that research is a very big part in combating AMR.
Yes.
Maybe you could share some of the research breakthroughs that have significantly influenced our strategies against AMR?
Yes. As I said, the rapid test kit is one of the breakthrough in any of the, not only the AMR, maybe the microbiological diagnosis. The rapid test kit is one of the breakthrough. But behind the rapid test kit, genomic sequence data is necessary. So now we can do the whole genome sequencing much cheaper compared to the previous days. So the whole genome sequencing is, I think it's the biggest breakthrough for this field and any other field as well.
So you can provide us answers that we're looking for.
Yes. But don't make our mistake for the whole genome sequencing data. It's not just the genomic sequence does not express the real phenotypes of biological characters of the bacteria or other microorganisms. So you have to see both on the genomic information as well as biological phenomenon or the pathogen. Then you can get the right answer or right the character or the specific bacteria.
I see. All right, thank you for that. So on a global scale, you mentioned that research can maybe help us with policies.
Yeah, it's
research findings. Yeah.
Yes, same as the answer for how the research helps to control the AMR spreading. It's same thing. The research evidence can support the policy making as well. So that's why the research is really essential. Yeah.
All right. Thank you for that. So that was Dr. Yukihiro Akeda. Is there a message you would like to give to our audience?
Yes, AMR is a really big problem in worldwide, and also the other type of the infectious diseases also the problem worldwide. So not only the AMR, but also the infectious disease globally. Then new techniques such as whole genome sequencing, rapid test kit, and other research evidences can help a lot in the field of the infectious diseases. So if you really like to help us, please join to this field. Thank you very much.
Yes, we need more young minds.
Yes, definitely.
Yes. All right. So thank you very much for tuning in to our podcast. I am your host, Vedika Bhatt. This AMR Wise podcast series is brought to you by University Malaya. This podcast series was created by Prasashila Ponampalavanar, an ID physician, and Associate Professor Dr. Cindy Tay, a microbiologist in University Malaya, and also me, myself, Vedika Bhatt. To learn more about us, please visit our website as mentioned below. The recording of this podcast was supported by the Department of Social and Preventative Medicine and the Visibility, Information and Communication Unit, University Malaya. Thank you for listening. Please join us and stay tuned for our next episode. This podcast is supported by a grant from the Foundation to Prevent Antibiotic Resistance. Till next time, bye-bye.